In prokaryotic and eukaryotic cells cytosol, the glycolysis occurs. The glycolysis does not require molecular oxygen, so it is common in both anaerobic and aerobic conditions. Thus, it is called as anaerobic glucose catabolism. The word glycolysis consists of two different words like glyco and lysis. The word glyco means sweet and lysis means split. So in glycolysis, the glucose is oxidized to two pyruvate molecules. The glycolysis is highly exergonic in the direction of pyruvate formation. For example, under typical intracellular conditions in our body, the delta G prime for overall pathway from glucose to pyruvate with the generation of ATP and 2-NADH is about minus 20 kilocalorie per mole. 10 water-soluble cytosolic enzymes take part in glycolysis. But in some parasitic protozoans called as trypanosomes, the first 7 enzymes are present in the membrane-bound organelles called glycosomes. All the reaction intermediates produced by these enzymes are water soluble and phosphorylated compounds are called as metabolic intermediates. The other name of glycolysis is Emp pathway, after the name of 3 German biochemists Gustav Embeden, Otto Meyerhof and Otto Paranus. In glycolysis, first the glucose is irreversibly converted into glucose 6-phosphate. In this conversion, 1 ATP forms 1 ADP. This is named as step 1 and is catalyzed by hexokinase. Then the glucose 6-phosphate is reversibly converted into fructose 6-phosphate. This step is named as step 2 and is catalyzed by enzyme phosphoglucoisomerase. The fructose 6-phosphate then irreversibly forms fructose 1,6-bisphosphate. In this reaction, 1 ATP forms 1 ADP. This step is named as step 3 and is catalyzed by phosphofructokinase 1. Then the fructose 1,6-bisphosphate reversibly forms 2 molecules of glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. This step is named as step 4 and is catalyzed by aldolase. The dihydroxyacetone phosphate reversibly form 1 glyceraldehyde 3-phosphate. This step is named as step 5 and is catalyzed by triose phosphate isomerase. Then 2 molecules of glyceraldehyde 3-phosphate reversibly forms 2 molecules of 1,3-bisphosphoglycerate. In this reaction, 2 Pi or inorganic phosphate and 2 NAD plus forms 2 NADH and H plus ions. This step is named as step 6 and is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Then 2 molecules of 1,3-bisphosphoglycerate reversibly forms 2 molecules of 3-phosphoglycerate. In this reaction, the two ADP molecules are phosphorylated to form two ATP molecules. This phosphorylation process is called as substrate level phosphorylation. This step is named as step 7 and is catalyzed by phosphoglycerate kinase. Now, the two molecules of 3-phosphoglycerate reversibly forms two molecules of 2-phosphoglycerate. This step is named as step 8 and is catalyzed by phosphoglyceromutase. Then the two molecules of 2-phosphoglycerate forms two molecules of phosphoenol pyruvate or PEP with the removal of a water molecule. This step is named as step 9 and is catalyzed by enolase. Then two molecules of PEP irreversibly form two molecules of pyruvate. In this reaction, two ADP molecules are phosphorylated to form two ATP molecules. This phosphorylation process is called as substrate level phosphorylation. This step is named as step 10 and is catalyzed by pyruvate kinase. These two pyruvate molecules are then converted into two molecules of acetyl coenzyme A, which will take part in two different TCA cycles. So, net gain of ATP in glycolysis is equal to 4 ATP minus 2 ATP is equal to 2 ATP. And the number of CO2 evolved in glycolysis is equal to 0. Please subscribe, like and share.